Okay, so we're back. Um, Going to kind of work to the second part of the cell structures, and we're going to get into just basically the cell membrane. Probably one of the biggest structures that we're going to locate. We're going to basically find within the cell, and mainly just because again that major theme, that major kind of idea that we talked about in the last video, which is just the homeostasis and the ability for that internal environment to remain stable, and because of the cell membrane, its ability to either to move substances in and out of the cell. We obviously have that capability of regulating the intake and the uptake of certain molecules. So pretty big structure in the case of a lot of, not even just a cell membrane, just a lot of the organelles that we're going to find within our cells themselves. Remember, we're not really talking about prokaryotic cells in the case of the organelles, but the eukaryotic cells. Any of these organelles also are the membrane-bound organelles. You're going to find the same type of materials inside of them. So a lot of things to kind of take into consideration. One, we're going to be looking at the movement of materials across the membrane. We also kind of start looking at these carbohydrate chains and being like receptors, chemical receptors or of hormones, how cells can communicate with each other, antibodies, little things like that. And so we'll kind of break that down. So a lot of different functions that we're looking at, but primarily we will be focusing on the whole transporting materials across the cell membrane. So probably the biggest thing we start looking at the membrane is just going to be the actual makeup itself. And you remember, or the parts that make it up. We've got what they call a phospholipid bilayer, right? And so you've got phosphate heads, we've got lipid tails, phosphate heads. We've got these carbohydrate chains, which basically kind of act as markers. And again, ways for cells to communicate with each other, or just as simply that whole shape determines function type of kind of theme that we looked at. We've got certain proteins. There's actually 4 or 5 different types of proteins that we're going to find embedded inside the membrane itself. And then we have cholesterol, which can also get thrown into the mix. A very, very important job as we go through. So some of the major characteristics we start looking at. Okay, phospholipid bilayer, selectively permeable, allows certain substances in, others not. Also has we coin a term amphipathic, meaning that it is both hydrophobic and it's both hydrophilic. Remember, the lipids we find on the inside are hydrophobic. They're scared of water, and that's kind of the driving force behind the cell membrane. And what gives us structure is that we've got water on the outside, we've got water on the inside. These little tails right here are so-called afraid of water, right? And so therefore what's going to happen is they're going to be driven on the inside of the membrane and kind of produce a little bit of structural integrity within that membrane itself, all right? So again, they are amphipathic, and then we've got the polar heads, non-polar tails, hydrophilic heads on the outside. So we kind of start looking at the membranes. As we just said, kind of the whole interaction of, the membrane itself, it's all held together by these hydrophobic interactions. It's all held together by basically the tails, so-called being scared or afraid of water. Now, when we also go back and take a look at these lipids, remember, there's 2 different types. We've got saturated versus unsaturated fats. And one of the differences between the unsaturated versus saturated fats is that double bond, which causes the tails of the unsaturated fats to actually kind of bend off to the side a little bit, like we're seeing right up here with these guys. So when we kind of start looking at the membrane, we want a membrane that's relatively fluid. But it varies. It varies in the time. It varies in the climate. It varies in the temperature range as far as what you're looking at. So you're going to see a lot of different varieties here as far as saturated versus unsaturated fats that you're going to locate inside the cell membrane itself, even at different times. And the one, we kind of start looking at, well, if completely made up of unsaturated fats, you're going to notice that these tails are kind of driving each of the molecules away. And so therefore, they're not as tightly packed together as you're going to see those with the saturated fats. And so this would create more of a fluid environment. And so if you take a look at these saturated fats over here, or membrane with complete saturated fats, again, nice straight tails, right? You're going to see that it actually has a larger amount of phospholipids in that area, which is going to make it more tighter in configuration, but actually make it more structured and less fluid. So there are some varieties. There are some reasons why. You take a look at individuals, you take a look at plants, maybe in the summertime. Temperature is already warm. Membrane, things are generally more fluid at that point in time anyway. Probably doesn't want to have a lot of unsaturated fats in that environment. We want something that actually can solidify and keeps the structural integrity of that membrane together. Same thing. You kind of get to the wintertime, you're going to find plants and other organisms that maybe have more unsaturated fats to try to keep that membrane a little bit more fluid in that environment. So again, you kind of have that individual breakdown of what type of lipids you're going to find on the inside and the fluidness of the membrane, and at what times is that going to be important to have something with a little bit more structure and something without. Cholesterol also comes into play with this. Cholesterol can actually have a factor of both tightening up or making that membrane a little bit more solid, or also can separate those molecules and make them a little bit more, in this case, a fluid. And so generally what you're going to see with cholesterol is that it's usually going to help to immobilize the membrane. In the case we talked about it kind of being a wedge. And so a wedge would basically, what's going to happen is it's going to force itself in one of these little areas. And eventually what it's going to do is it's just going to drive those molecules a little bit closer to each other. And it just makes things a little bit more tightly compacted, right, to a point to where they just keep tightening up. And so generally is going to make things a little bit more solidified. Now, also during sometimes of the year, that cholesterol can actually come into play, because as it's driving, some of those other molecules together, what it's also doing is it's also separating molecules. And so in a time where maybe it wants it to be a little bit more fluid, right, that cholesterol can actually separate those molecules and keep a little bit more fluidness within the membrane itself. So cholesterol does have kind of I guess two different forms, two different ways that it can help out. Generally, it's going to be immobilization of that membrane, though. So cholesterol, obviously, it's good in moderation. We don't want to build up too much of that, but we do want a little bit for our membranes to make sure that they are in good standing. Okay, but it's always good to have differences in those compositions just to make sure that again, you can kind of make up for those fluctuating temperatures. And I think the example we talked about in the past was winter wheat and how they can change the saturated versus unsaturated fats just on based on different times of the year. So the other part that we start looking at, the carbohydrates, obviously a big part in cell surfacing, or the markers. We'll talk about those with the communication coming up, but going to be the proteins. And the proteins, there's actually 5 different kind of functions that we're going to find with the proteins embedded in the membrane itself. And probably one of the major ones is going to be transport. And that's just basically aiding in molecules that are either too big or they're polar. They can't get through that hydrophobic interior, that lipid interior. And so therefore they need some form of membrane in order to get through. So something, water is not a great example of that, because it does have channels, but they're called aquaporons. But you're going to find, salt, sugar, things like that, that can't get through. They're going to need to basically go through those proteins. Enzymatic activity, obviously. Okay, we know proteins make up enzymes, or enzymes are made up of proteins. And so therefore we're going to find some of these enzymes that are going to be located in the membrane itself, and basically will be marked, or can simply change shape and become activated by certain hormones that you're going to find floating throughout the body, and as they come in contact. Signal transduction, shape to read a specific hormone, just kind of went through that a little bit, but this is more about the situation of, actually, when we go back to 2 is more about the organelles themselves. And so every organelle is going to have different enzymes when you kind of start looking at different types of enzymes in order for it to function. The signal transduction is more about the cell communication with each other, and how we have, again, specific hormones that will basically reach the membrane. They can't fit through. So they're probably polar. Maybe they're, a situation, whatever, they're too large, they can't get through. So what they're going to do is they're going to activate an enzyme, right? Activate one of these proteins that we're going to find in the membrane, which will then activate, another enzyme inside the cell, or send signals to other parts throughout the cell and such. So it's a way for cells to communicate with each other as well. Cell-to-cell binding, antibodies. Antibodies are an example of a protein. Antibodies would be an example of where we can mark viruses and bacteria for destruction. If something is foreign to our body that we don't really want or need, we think that it basically is going to put us in danger. It's a situation to where we can mark it, and then eventually we'll come back and kill it off, or basically destroy it. We didn't really get to the immune system all that much this year. It was going to be something that we were going to cover after we get in with ecology. But again, the human body itself is not really all that important, but just kind of understanding the overall process of we've got cells that we don't want. We mark them for death. Eventually we come back and we destroy them. We got macrophages, which would be types of cells that are going to allow us to do that. And then We've got also the proteins, which are basically the attachment of the cytoskeleton. Cytoskeleton is what kind of gives the cell structure and support. And so therefore, again, it's not like a balloon to where if you just let the air out of it, all of a sudden that balloon just shrivels up and shrivels up and shrivels up. Is that we have cells that obviously can lose water. They can lose that fluidity inside of it, but yet they're still going to maintain some of the structure because of that cytoskeleton that you're going to find basically kind of working to the cell membrane and eventually attaching to that cell membrane and providing it with strength itself. Okay. So we go through, take a look at carbs again, cell to cell, recognition, kind of just ways for cells to understand, what is good and what is bad and such. So as we go through again, right, we kind of start looking at where we're going to go from this as far as the membrane goes. We're going to kind of carry this into the passive versus active transport. We're going to kind of start looking at, just the what, the membrane does in the ability of moving substances across. I guess one of the other ones that I should go back and re-hit a little bit is that the membrane itself also, when you find them around some of these very specific organelles inside the cells, it provides an environment for those cells to work individually and and also work as a team, because obviously it's in the cell itself, but it gives it its little own environment to where, the mitochondria can do what it needs to do. The endoplasmic reticulum can do what it needs to do. The ribosome is a bad example because they don't have membrane, but some of those, and so it gives them that environment just to function the way that they need to, provide them with the certain enzymes that they need to function as well. So again, we'll kind of jump into the cell transport here, passive active transport, eventually jumping into water potential, and then we'll be finished up with the cell structure unit.